So thank you, Philippe. I just want to add, so Cy Leventhal was my mentor. As most of you probably don't know his name, but he was responsible for all computer graphics in biology. And it's embarrassing to say, but I was a postdoc with him in 1970, and the graphics system, so most of you weren't even, maybe your parents weren't born. But anyway, it really, he had a interactive computer graphics system built, which was literally the best system I've ever worked with. And if iCn3D can reproduce even some of that, it would be a good thing. But that's not the topic of my talk, so I'll and is there a pointer, or should I just use the arrow?
Use the mouse.
The mouse, okay. Okay, so I work on protein-protein interactions in very different settings. So on the upper left, it represents a major interest of my lab, which is understanding the role of adhesion proteins in the development of the nervous system. And basically, cells recognize each other, neurons know who to synapse with, and it all comes down to what proteins are on the surface and what they recognize. And the way we attack this problem is through crystallography, measurements of binding affinities, and the design of mutant proteins that can affect how the nervous system is wired. So it really brings molecules to the level of neurobiology. We also have a project, or a series of projects, using molecular dynamics, free energy perturbation methods to study the effects of mutations. And both of these areas I think of as old-fashioned, low-resolution use of structure. We care about every angstrom, we care about every kilocalorie. And I still do, but over time, in part inspired by the Structural Genomics Initiative of the NIH, which has been defunct for many years, we became interested in using structure to predict whether two proteins interact. And we found a way to do this on a proteome-wide scale. So some of this work is published, but I'm going to go over it and sort of bring us to where we are today. The ultimate sort of how do I flip slides?
Just use that the cursor. This one?
This one?
It doesn't work.
I'm sorry?
So you keep the mouse here. Look at the screen.
Okay, but how do I get to the next slide? What did you do?
Just next, because it was on the screen. Keep the cursor here.
Okay.
And then, because it was on the screen, so you're pressing next.
Next, okay.
So,
All right, let's try it. Great. So this is a sort of an outline of the PrEPI algorithm. And for those of you that are familiar with it, it's not pre-PPI, it's PrEPI, which has a second meaning. Anyway, we start with two proteins. If we know their structures, we build models of those structures, and now we simply use AlphaFold. And here's models of the green and yellow protein. We look for structural neighbors using structural alignment, and we look for those neighbors in PDB complexes. If we find such neighbors, then we effectively build a homology model of acting with the green. And next is this. Yeah, I think I'll take my glasses off for this one. This is just an example of a model that we built. The brown and purple proteins are from Arabidopsis. The yellow and green are models we built for two human proteins. And this is a relationship that you couldn't find from sequence or not easily, but it's a structural relationship. Now, what I'm going to tell you in a minute, but now is that we build models, we build billions of models. And we have to evaluate them and get a score that tells us whether they're likely or not likely. The question is, how do we do it? We obviously can't do a simulation on billions of models. So what we do is the following. This is sort of a schematic of how we evaluate models. This would be a PDB structure. and we know what residues are in the interface, and this we simply do for the whole PDB. We now superimpose our proteins, and we get a score based on how good the superposition is. The real trick is we now simply ask how many of the contacting residues, in the template are present in our model. And then we ask how many of the contacting residues in the model are predicted to be interfacial. So we get a score, we measure each of these variables, and because we're only looking at a few interactions or a few residues in an interface, then the evaluation is immediate. We're just really counting interactions, which is what makes it very fast. And when we're through, we get what we call SM, a structural modeling score. To this, we add lots of other information. So if we have a model, some of our models are probably very bad. Some of them are good. We're going to add more information. We add information. Are they co-expressed, for example? Is there a sequence relationship between them, et cetera, et cetera. So we take all of this information from different databases, and we have to train each of the scores that we get so that we have some way of combining them. And to do this, we simply have positive and negative databases, and I'll say a bit more about that in a minute. And we use Bayesian statistics. If there's a parameter X which is enriched in the positive database relative to the negative database, we get a likelihood ratio. And the nice thing about naive Bayesian statistics is that you can simply multiply likelihood ratios. So we can get likelihood ratio from structure, we can get likelihood ratio from co-expression, et cetera, et cetera. An issue that comes up in this whole field is how do you evaluate with what your predictions? And there are many, many databases of protein-protein interactions out there. And this just takes a few of them, and there's a Venn diagram. And the main point here is to show you how poorly they overlap. It's really a problem. Another issue is that not all of them define an interaction in the same way. So in terms of predicting structurally, we're interested in two proteins that physically are in contact. Well, a lot of the databases say two proteins interact if they have a functional relationship. They might be in the same pathway. So we wouldn't want to train on such a database if all we're doing is looking at physical interactions. We use a database called HINT, high quality literature curated from Haiwan Yu and Cornell. They put in a great deal of effort in defining binary interactions. They also have other less precise databases. And we train on HINT. We could train on the PDB. HINT has lots of PDB structures, but it also has interactions that are not in the PDB, so it sort of gives us more breadth. To show you how we perform, I have area under the rock curve calculated for a series of different databases. And with HINT, we do tenfold cross-validation. And the structural modeling score, this is beyond my, getting close. Our area under the curve is 0.83, but now if we use the training of human, we're training on human, and predict yeast and E. coli from HINT, you see we're getting similar performance. So we're not really degrading, in fact, we're getting better with E. coli. Now, if we add all the data, all the non-structural information, that's what all means, the performance on HINT degrades because it's introducing information that's extraneous to what we're trying to do. I could go through all of this. I don't want to because of time, but what you can see is we do terribly on some databases, especially if we're only looking at structure. For example, for BioGrid, it's not really very good. If we add non-structural information for BioGrid, we get much better. Many of BioGrid's interactions are indirect. So I'm just pointing this out as a problem. It's very hard to evaluate how good you are, and it's very hard to know what database you're using to evaluate yourself. And this problem hasn't gone away, but I do want to make the point. Right now, we have a database for human on our website. Most of the proteins are represented. Where does the number billions come from? Well, we have about 20,000 proteins with AlphaFold models. We break each protein up into domains, so we can treat each domain separately. And that gives us a total of 95,000 or so full-length proteins. And by the way, divide into CDD domains for some of the people in the audience. This would lead to 4.5 billion possible interactions. But then we take more than one model. There may be more than one template in the PDB. We pick the best one. So we're up in the range of 20 to 30 billion evaluations that we have to make. And we think about 1.3 million are on our database, and we think about 400,000 of those are direct. Database is available for the whole data for download, or you can interactively look at some of these structures. Yeah, here. You can type in protein, you see its neighbors, you can look at some pictures. It's fairly primitive, and I think interacting with a more refined viewer would be very nice. So this is what PREPI is. There are different sorts of evaluations that we do. One of them is simply what I said, looking at rock curves on different databases. We've tested some of our predictions experimentally. And we're constantly refining it and improving it. And a recent paper of ours, we have something we now call ZEPI. If we have a protein structure, and these are the interfacial contexts, we calculate a series of variables. One is coevolution with mutual information. One is simply sequence conservation on either side of the interface, and we have a few others. And we create 1,000 fake interfaces. We have one real one, and we get a z-score for how well our interface compares to the others. This is a very messy slide, so I'm just going to say here that we look at the performance of each of these variables, and then in any one case, we simply pick the best score. It may come from coevolution, it may come from sequence conservation, and that's the ZEPI. The point, actually, I should have, want to make is that we don't have to worry about three body effects. So people use, for example, direct coupling analysis. If you're taking an entire sequence and you want to make predictions, you have to worry about indirect interactions. We don't have that problem because we already know what interface we want to evaluate. We simply want to see if there's coevolution there or not. And that means we can use very shallow multiple sequence alignments, and even as low as 10, we get signals. So it makes it easier to get paired multiple sequence alignments when looking at, when evaluating structures. We've added ZEPI to PREPI as a filtering device, and I'm not going to go through this busy slide except to show that if we have a low false positive rate here for human and a good ZEPI z-score, we find on the order of 3,000 novel interactions to find as appearing in no other database. And we've looked at some of those. And just to give you a sense of sort of going to low resolution, there are two proteins involved in synapse formation, sharpen and shank1. And we've been studying KRAS because of its cancer relationship, but we found that KRAS interacts with both sharpen and shank1. And looking at the literature, it turns out that, believe it or not, KRAS plays a role in inhibiting or activating synapses. So it doesn't prove that we're right, but it is what we're really interested in with PREPI is hypothesis generation that allows us, if you're interested in the biology of the problem, to dig deeply and look into it. How am I doing?
Oh, you're doing very well.
That's unusual. That's great. Okay, great. So I'm going to go through two applications. This is just to say one of the things we're doing with PREPI now is instead of just using ZEPI as a filter, we're calculating likelihood ratios from ZEPI. We've also started interacting with the Cowan-Berger group, trying to integrate, in this case, language models as an additional source of evidence. This slide I got last night, so I won't vouch for it, but what it appears is that the big effect of combining PrEP with topsy-turvy, a language model from the Berger lab, is that we improve the precision recall curve dramatically by combining them. So, but we're still working on this. But the nice thing about using Bayesian statistics is you can always introduce another source of evidence. So we can start combining structure with AI based with deep learning, which you better be doing these days. So p-hipster, just where the name comes from, some of you may know that preppy is a personality type. Sagi Shapiro, when he was at Columbia, started working with me as a virologist, and the way Sagi dresses, he wasn't comfortable with preppy, so he called it p-hipster, which is pathogen host something or other. And what we do is we simply use the preppy pipeline, but now looking at virus-human interactions. And we looked at all human infecting viruses, and at the time there were 1,000 in the database we looked at, 12,000 proteins. We tried to build homology models. This was, even today, it's hard getting AlphaFold homology models for virus proteins, but I think that field's getting better. But anyway, we built our own. We have models for human protein. We ended up with 280,000 predictions that made sense to us. And we applied, so we now have an interactome between 1,000 viruses and human, and we looked at some special cases, and I don't have time to talk about all of them. One thing we did was he actually measured, tested some of our predictions with co-IP, which isn't the best way to measure, but you can see that ratios means a positive result, blue, and low likelihood ratios means that there were some bad predictions. But overall, the sensitivity and specificity we found were about 75%. So these were experimental tests on some of our predictions. So obviously they're not all right. It would be foolish to think that they would be, but we feel they're good enough to start thinking, perhaps intuitively, along the lines that Philippe was talking about. And here's just one application. It's a messy slide. So on the right are human papillomavirus, different numbers. And on top are human proteins predicted to interact with papillomaviruses. The orange ones are those that are high risk, they cause cancer. And you can see, and the blue ones don't, you can see that what we call type 3 interactions are present in both the high and low risk, but there's a preference for the high risk papillomaviruses to interact with some human proteins, and a preference for the low risk interaction, low risk viruses to have interactions with different proteins. A lot of what we found here was known from sequence. So this selection of a high risk, low risk isn't new. What's new is that we could do this by predicting protein-protein interactions. And it gives you a much better picture of what might be going on. It allows you to look at pathways, et cetera, infection pathways, in ways that you couldn't otherwise do. So finally, there's one other application I want to talk about. We work with Andrea Califano, a colleague in my department, and Andrea looks at protein-protein interactions in a very different way. These are breast cancer, actually lung cancer samples from the NCI, and there are going to be horizontally 20,000 genes and vertically 500 or so patients. And basically what they do is all these proteins or RNAs are sequenced. Blue means upregulated, red means down, actually red is up, blue is down. And they know the mutation state of each of these. So you can begin to say, if this protein is mutated, what other proteins are affected? So you begin to get regulatory interactions from patient data. That's what Andrea does, but they don't have information about direct physical interactions. So these various methods on the right are Andrea's methods. On the left is PREPI. We find them using a random forest approach and can focus on a particular protein of interest. In this case, we focused on KRAS and came up with a network of interactions centered on KRAS. Some of these have structures, some of these we have predicted structures. We know which ones are druggable. I'm obviously going through this quickly because I just want to give you a flavor of what can be done. And we found a collaborator, Alejandro Sweet Cordero in Stanford. And what these guys do is they give mice cancer. And so they infect the mouse with mutant KRAS. They eventually break up the cells. And then they use RNA knockdowns to inhibit different proteins that we predicted would be interacting with KRAS. And eventually you have a list. These are proteins, oh God. On the right, the figure on the right shows you proteins that when you knock them down, cell proliferation, the tumor doesn't grow. And red are our predictions, green and purple are various controls. It looks actually worse than it is, and it looks pretty good because every experiment is done 4 times. So it's an average, but our predictions turned out to be right 18 out of 21 times. So those 18 out of 21 then provide a hint of what might be interacting with KRAS. It allows you to look back at pathways and start thinking intuitively about what might be going on. More generally, there are methods today, genetic interaction methods, where you can see whether 2 proteins work together at the gene level. It doesn't mean they're touching again, it just means are they working together? And what we're beginning to do now is integrate PREPI with these methods to come up with predictions of proteins that play a role in this particular set of interactions. We could come up with maps of interaction with networks and then actually build models of what might be going on there. So possibility of applications of this type becomes real if you have a method that can work on any pair of proteins you're interested in, and that's where PREPI comes in. I want to emphasize again that we're interested in hypothesis generation. I think I'll stop here and take questions, but I do want to emphasize that you want to think like a biologist or someone interested in a particular set of problems, then development of hypotheses, getting at proteins, getting at mechanism, allows you to think about these problems in the traditional way, but getting there requires new high throughput methods, both computational and experimental. So thanks for your attention.
I have a couple of questions, but I'll ask only one in the interest of time. So what is the difference between PREPI and AlphaFold multimer option? And how does it scale in terms of computing the interactions?
Well, if I have any pair of proteins and I want to predict how they interact, I would use AlphaFold multimer. If I want to look at 1 billion interactions, I can't. So the scale, we can run the human proteome in about a week now for literally looking and evaluating 30 or so billion interactions. So I view this as hypothesis generation, screening device. If you then become interested in some particular problem, you're going to turn on the best methods available for structure prediction. And PREPI, if we've done some tests, if the homology is good, then we get models very similar to AlphaFold multimers, sometimes even better. But when you get to more distant models, then our models capture something of the interface, but are not accurate. So it depends where you stand.
Thank you.
It's all a matter of scale, and I should have said that.
Thank you.
Hi, Sara Capponi from IBM Almaden Research Center. Very great talk. So this is a very naive question. So during the talk, you show the interaction mostly between KRAS and the other proteins. So I was wondering if PrEP works also for membrane protein interaction.
Yes, it does.
Okay.
I didn't think it would, by the way, but it's giving us, it's behaving quite well. We're actually working on that because we're using PrEP. Well, it works for membrane receptors very well.
Mm-hmm.
So, but most of those are stuck in the membrane but are in solution.
Yeah.
So I have to take what I said back. For receptors, yes. For peripheral membrane proteins, yes. We haven't really looked at whether it's going to pack alpha helices in the bilayer. So I can't answer. So that I don't know.
Okay. Okay. Yeah. Thank you very much.
It's really low, but that was fun. Thank you so much. My question is more along the lines of an earlier question. What is the scale? So I'm interested in emergent interactions in the microbiome world. So if I were able to pick out 10,000 genomes and predict the proteins and predict their structures, what can I expect in terms of performance and in terms of time it would take me to predict the interactions?
Well, if you're dealing with viruses, and I'll get to bacteria.
Yeah.
The nice thing about viruses, they don't have most of them don't have large genomes.
Right.
So the scale would be similar to doing one large eukaryotic genome in terms of time. We haven't yet done bacteria human, but we're beginning to work on bacteria. I don't think scale is a problem. Eventually, obviously, if you want to run tens of thousands of bacteria, it's a problem. What we're trying to produce now, and it's hateful for me, but it's producing code that we can simply give to everyone and say, run your own system. Right now, we have to do it. There's just too many proscripts. But one day you'll be able to do it if you're interested, on your own system.
Sure, but I'm most interested in the interactions of the microbes within the microbiome, not microbes.
Oh yeah, so we just have to run it. It's not a problem.
So you don't need all of this additional information that
Well, that's the thing. We don't necessarily, issues like co-expression. So if we're just looking at an individual bacteria, then we're going to start with just structural. What are the structural predictions?
Okay.
So in a way, it's easier, but you're losing information unless it's out there. Go annotation we use. It's most single effective non-structural evidence we have is simply go.
I'm sorry, very rich.
No, it's okay.
Been to the house session this afternoon.